Hello, everybody. Today we're going to talk about replication of medium DNA viruses, talking about single stranded DNA viruses. Uh, I am going over lecture uh, slides set number 11. So, slide number 2. The family of small DNA viruses, single-stranded DNA that we are talking about are adenoviridae. Adeno means gland or lymphoid tissues in nasopharynx. These viruses have icosahedral capsid, 70 to 90 nanometer. They have fiber projections that they project out of the cell, the virion, on the surface of capsid, and they serve as ligand for attachment. Some of these adenoviridae family members cause cancer or tumors in experimental animal models, usually the newborn hamsters. Moving to slide number 3, the first genus is Mastadenovirus, mast, breast. It is specific for mammalian species. Human adenovirus 1 through 51, these are the serotypes. The host range is pretty wide. It can infect monkeys, horses, cows, pigs, and dogs. Slide number 4. Another genus is aviadenovirus, AV, bird. It is specific for avian viruses. Slide number 5, human adenovirus. In 1953, when researchers were looking for common cold virus in tonsils and adenoid tissue cultures, they discovered this virus. Latent virus can become activated, it can replicate and causes CPE or cytopathic effect of the host cells. Slide number 6, human adenovirus infections. The main infection caused by human adenovirus, the main infections are URTI and LRTI, upper and lower respiratory tract infections. It can also cause epidemic conjunctivitis of the eye, gastroenteritis, inflammation of the GI tract, and can cause acute hemorrhagic cystitis, which is bleeding, acute bleeding from the urinary bladder, the muscular sac that serves as a reservoir for urine. The patterns of adenovirus infection are lytic, chronic, latent, and they can also transform cells. Upper respiratory tract infection, slide number 7, or simply called as common cold, is caused by a variety of viruses. The main culprit in causing cold is rhinovirus or rhinovirus, followed by coronavirus. Adenovirus is in the third place, and then we have parainfluenza virus. So as you can see, we get the cold, but the causative agent can be a variety of viruses. Moving to slide number 8, upper respiratory tract infection, pharyngitis. Adenovirus is the main common cause of pharyngitis. Other viruses that can cause pharyngitis are herpes simplex virus, enterovirus, Epstein-Barr virus, or EBV, and those viruses that are less likely to cause pharyngitis are influenza, parainfluenza virus, rhinovirus, and coronavirus. Slide number 9 is a little bit of a description of pharyngitis, which is, ITS at the end, inflammation, and pharynx or throat is inflammation of the back of the throat or pharynx. Slide number 10, upper respiratory tract infection, lower respiratory tract infection, or CROP. We had discussed this illness before. CROP is also called laryngeotracheobronchitis. Laryngeotracheobronchitis. The causative agents are parainfluenza as number 1 and RSV or respiratory syncytial virus as the second causative agent. Moving to slide number 10, I have included a little bit of a definition of CROP or laryngeotracheobronchitis. Moving to slide number 12, lower respiratory tract infection, bronchiolitis, which is the inflammation of the bronchioles. Parainfluenza virus, number 1, and respiratory syncytial virus or RSV as a second prominent causative agent. So as you can see, these two viruses were also capable of causing CROP, as we discussed on slide number 10. Moving to slide number 13, lower respiratory tract infection or pneumonia, infection of the lungs. Influenza is the first common cause, followed by respiratory syncytial virus, RSV, parainfluenza, adenovirus, and coronavirus. Moving to slide number 14, this is a list of some of the cold virus symptoms, the symptoms that patients exhibit upon being contracted with the cold virus. These include fever, cough, headache, sore throat, muscle ache, fatigue, and presence or absence of vomiting and diarrhea. Moving to slide number 15. So if you have some issues that you think, oh, I may have a cold, but I'm not really sure, you need to see a clinician. If you have a fever that is greater than 130 degrees Fahrenheit, or a fever of 101 degrees Fahrenheit that lasts for more than 3 days, yellow or green discharge from lungs, extreme difficulty swallowing, shortness of breath, chest pains, earache, and discharge from ears, and in some instances, loss of hearing. If you have these symptoms, you definitely need to see a clinician. Moving to slide number 16, let's talk a little bit about the organization of adenovirus genome. As I mentioned, is a double-stranded DNA virus. Adenovirus type 2 has a linear genome, 36 kilobase per. Ends have something called inverted terminal repeats. These are single-stranded DNA sequences that can form circular structures because of complementarity base pairing. The repeats at the end of this strand are inverted, so they're very similar. The 5' end of the genome, we have a protein, a 50 kilodalton protein called viral terminal binding protein, or TBP. TBP comes from a precursor protein that is 80 kilodalton, and this 80 kilodalton precursor protein serves as a primer for DNA synthesis. Genes can be found on both strands, both DNA strands, and upon gene expression and translation, they express proteins that code for cellular activation, DNA synthesis, suppression of host immune response and inflammatory response, as well as structural capsid proteins. Inflammation, there's always a question whether inflammation is good or bad. Inflammation, depending on the duration of it. Of course, if we have an infection, inflammatory response is a natural body and immune system's response to curb the dissemination of the infection. However, if it lasts for long periods of time, it is detrimental to the body. Moving to slide number 17, adenovirus early messenger RNA. Early gene expression is done from both DNA strands using host cell RNA polymerase II. We have right strand transcription and left strand, or positive and negative sense, or antisense, whatever you want to call it. And overall, we have 4 early transcription units, E for early, E1 through E4. Each one of these transcription units have their own promoter, those sequences that get activated and promote the expression of the gene. And E1 unit has an enhanced promoter that is also gets activated by host cell DNA binding proteins. So there are proteins in the host cell that bind DNA. Upon binding, those proteins activate the promoter for E1 transcription unit. The messenger RNAs produced have 5' 7-methylguanine cap and 3' poly A tail, and after they are being made, or after transcription, they will be spliced to make a group of proteins. So after transcription, we call it post-transcriptionally spliced. Moving to slide number 18. What are some of these adenovirus early proteins? There are 4 proteins that we'll discuss. The first one is E1A, E1B, that has transformation function, meaning that its expression leads to transformation of the host cell. How does it do that? By binding and blocking cell cycle repressor proteins, P105RB and P53. We talked about these two, guardian of the genome. P53 and PRB regulates cell cycle progression. Another protein made early on during adenoviral transcription is E2. It's a DNA polymerase, and it's also a precursor to terminal binding protein, or TBP. It is also a DNA binding protein, meaning that it has a protein that has a consensus sequence or a unique sequence on the DNA that it binds to. And the function is to shut off early promoter, so serves as a negative feedback. E3 is the next early protein encoded by adenovirus that decreases host MHC proteins or major histocompatibility protein. These are basically surface structures that serve as they serve to present peptides to immune cells. So imagine if an immune cell cannot recognize viral proteins, we do not have an immune response. Subsequently, inflammation shuts down, and another protein, E4, also participates in decreasing host cell T cell recognition. So without going too much into the details of how T cells recognizes MHC, just know that T cells recognize MHC by a structure on their surface called T cell receptor, and T cell receptor recognizes a complex on the surface of the cells called peptide MHC complex. If the MHC is downregulated, as is also seen very frequently in HIV during HIV infection, host cell T cells cannot recognize, and the infected cells will evade immune response and continue growing and dividing. Moving to slide number 19. So one of the mechanisms by which adenovirus E1A/E1B protein activates host cell is via RB inactivation, whereby this protein E1A/E1B binds to P105 RB protein. RB no longer functions to regulate cell cycle or cellular divisions, and cells will continuously undergo cell cycle and proliferation. Remember, a couple lectures ago, we talked about RB, that there are these two transcriptional activators, DP and E2F, that RB binds to them to regulate them. RB gets phosphorylated and inactive. So one of the functions of this viral protein E1A/E1B is to come and basically sequester RB, take it away from the environment, so E2F and DP are constitutively active, and there is an unchecked cell cycle going on in the virally infected cells. Moving to slide number 20. Another mechanism by which E1A, E1B adenoviral protein inactivates host cell is by binding to and inactivating P53. Therefore, P53 can no longer suppress cellular division. Cells remain in an active state whereby there is no regulation of cell cycle, and the cell cycle keeps going on. The cells undergo unchecked proliferation. Moving to slide number 21, adenovirus DNA replication. Adenovirus has a double-stranded DNA, therefore, continuous DNA synthesis can potentially occur at either end of the genome. In order for the replication to proceed, we need a replication complex that is made up of a DNA polymerase that virus brings in with itself, so a viral DNA polymerase, a terminal binding protein or TBP precursor, the 80 kilodalton protein, as depicted by an oval bluish, purplish color at the 5' ends of the genome on the top figure. This 80 kilodalton protein, or TBP, serves as primer where DNA synthesis starts at a bound cytosine. And also we need a group of DNA binding proteins, those proteins that belong to either virus and or the host cell, and they bind to DNA. They serve as transcription factors, they serve as activators, recruiters, chromatin remodeling. You don't need to worry about too much the details of it. Know that the adenovirus DNA replication can use type 1 and type 2 models of replication. We will talk about these two models in subsequent slides. Moving to slide number 22, let's talk about type 1 DNA replication of adenovirus. So DNA synthesis initiates at the 3' end of one of the strands. So if we have one strand that we make a copy of it, we end up with a double-stranded genome, the parent strand and a new strand, and we have the original parent strand number 2 that will be displaced by single-stranded DNAs. So what will happen is these single-strand DNAs will accumulate in the cells. These are intermediates, and due to inverted repeats that are at the end of the sequences, they form circular single-stranded DNA structures. Okay, let's do it again. We have two strands, parental strands. In type 1 adenovirus DNA replication, we make a copy of one of the strands. So we have the result is a double-stranded. One strand is the old strand, and another one is a newly synthesized strand. Synthesis, the direction of synthesis is 3' to 5'. So we have a double strand, and the parent second strand that will be that is there a single strand. So it's a single strand DNA is inside the cell. Because of the terminal inverted repeats, they base pair, so we will see a double-stranded. This double strand, one strand is old, one from the parent, one strand is newly synthesized, and a bunch of single-stranded DNA structures. Whereas moving to slide number 23, whereas in the adenovirus DNA type 2 replication, type 2 replication takes place from either viral genome. So we have the 3' end that is on one strand that can serve as a template. We make a new strand off of it, and then we have 3' end of the second parental strand that we make DNA off of that. So the two parental strands will serve as templates, and we can make new strands. These two new strands that are being made, we end up with two double-stranded. One of the strands is from the parent in blue, and the other one is the newly synthesized strand in orangish pinkish color. Another way of doing it is to have those single-stranded DNAs that we made in the during type 1 replication. We can displace them and make them double-stranded, so they can serve as as template to make additional strand. So single-stranded DNA is being copied into double-stranded genome. It cleaves the 80 kilodalton precursor to 50 kilodalton terminal binding protein. So why don't we go back to slide number 22 again to go over this? The adenovirus DNA is double-stranded DNA. There are two types of replication. One type of replication always goes 3' to 5'. That's the direction. Okay, so during type 1 replication, one of the parental strands in blue will open up, serve as a template. At the bound cytosine, we have TBP in blue. It will come, and it's the primer, so DNA synthesis goes on. What we end up with is a double-stranded DNA. One is in the middle of the diagram. One strand is 3', 5' in blue. The other one is 5' to 3' in purple, in pink or orange. That's the newly synthesized strand. The second strand, the second parental strand, or strand number 2, just hangs out there. There's no replication going on from that strand. So we end up with a bunch of those accumulating as intermediates inside of the cell. Initially, they're linear, but due to terminal inverted repeat base pairing, they will circularize. So we see a lot of single-stranded, circular single-stranded DNA structure. Moving back to slide 23, type 2 DNA replication in adenovirus can be done in two ways. One is both parental strands in blue will serve as template, 3' to 5' directions. We get new DNA strand synthesis going on, or we can use the single-stranded DNAs that we have generated during type 1 replication. Those can be served as template, so we make a complementary copy to that. We end up with double-stranded DNA genome. Moving to slide number 24, this is basically a depiction of what we just talked about. One major issue that I want you guys to all remember and understand it is that during the replication, adenoviral DNA replication, we do not have any discontinuous strand synthesis. So all is continuous. The only difference that is very unusual in eukaryotic DNA replication is that there would be an accumulation of large amounts of single-stranded DNA molecules that will later on circularize due to terminal inverted repeats. Moving to slide 25. So far we've had early gene expression, genome replication, type 1, type 2. Now we have late gene expression due to the production of or transcription of late messenger RNA. It starts from one strand, one of the two DNA strands. It's the right strand transcription, left to right. It has a single promoter, that the promoter has a leader sequence and codes for 5 messenger RNAs, L1 through L5, leader 1 through 5, by alternative splicing. The gene products or the proteins that are being translated from these messenger RNAs are mainly structural proteins. Moving to slide number 26 is just basically the genetic and transcription map of adenovirus and some of the genes. You don't need to worry too much about this slide. So as I just mentioned, we go through this alternative splicing. We have single promoter that has leader, and we make 5 messenger RNA. So this messenger RNA is a result of mRNA splicing. mRNA splicing, the first observation for mRNA splicing was done by studies of mapping mRNA on adenovirus DNA genome. And they noticed that a late hexon mRNA, it's just called hexon mRNA, hybridizes with single-strand viral DNA. And this mRNA, which we call it hexon, and DNA complex, the duplex, is not collinear, and it forms single-stranded DNA loops. So why it makes loop if they hybridize? The simplest explanation and the logical explanation is that DNA is longer and messenger RNA is shorter. So the messenger RNA is shorter because it only has exons 1, 2, 3 leader sequence and a hexon sequence, and it lacks certain DNA regions. We call them introns, A, B, C. So they found that this messenger RNA, hexon messenger RNA, hybridizes with single-stranded DNA, but it's not collinear, and it forms loops. The reason is that either the DNA is longer or the RNA is shorter. We found that the RNA is shorter because it only contains exons and it does not have introns A, B, C. Moving to slide number 28. Adenovirus, we have virus-associated RNA. What is this? It's an RNA that is being synthesized by host cell RNA polymerase 3. These are small or short viral RNA molecules. And what do? They block the action of interferon. Remember, we said that viral infection leads to production of interferon. Interferon binds to interferon receptor, activates the transcription and translation of antiviral effector molecules, one of which is protein kinase R. So these virus-associated RNA molecules block the activation of protein kinase R. Also, another advantageous tool of virus, it's the presence of E1A protein that also, along with virus-associated RNA molecules, block interferon signal transduction. So when you think about it, virus uses multiple mechanisms to hamper or block local immune response, which is the interferon signaling and production of antiviral effector molecules. The result is persistent adenovirus infection. Moving to slide number 29, adenoviral can be used as a vector for gene replacement therapy. Bottom line, we use adenovirus because it has genes in it. We replace some of these genes, put it into patients. See how it works. We eliminate and cut out that gene segment of the virus that confers pathogenicity to the virus. So the virus pathogenic gene is being eliminated. We cut it out. We map it. We know where it is, which sequences. Then we use restriction enzymes like scissors at both ends of the gene, cut it, take it out, and we replace that with a therapeutic gene or gene of interest. We insert it back, we ligate it back into the adenoviral genome that is now devoid of the pathogenic gene but has a therapeutic gene. Example would be insulin gene. Then we infect patients with this adenovirus vector. So once the vector gets in, starts producing insulin and will be targeted to pancreas. Beautiful, correct? So far, so good. We have a bad gene in the virus. We take it out, we put the good gene, and hopefully patients will benefit from that. However, there are some pros and cons to this. The disadvantage is that sometimes we have complications because adenovirus infects other organs. This is called random insertion. It gets incorporated into other tissues and organs. Therefore, it causes side effects, results in toxicity, which is obviously not good for the patient. However, the very, very good point about adenovirus gene therapy protocol is that the adenovirus DNA does not integrate into host cell DNA. That's beautiful, because if it gets in there, as long as the host cell is replicating, the genes under the control of the vector or the genes inserted in the vector will also be producing, which is not very advantageous. The good thing is that this virus will eventually get degraded by the host cell. Moving to slide number 30, the next family is Parvoviridae. These are small single-stranded DNA molecules, icosahedral capsid, significantly smaller than adenovirus, 18 to 26 nanometer, as opposed to almost 70 nanometer of adenoviruses. The genus that we talk about is parvovirus, uses humans, mammals, and birds as its host, and causes a non-defective virus, so it doesn't cause infections. We'll talk about that in a minute. Another genus is Dependovirus. The virus in this group is called adenovirus-associated virus, a virus that is associated with adenovirus. So it requires help from adenovirus for replication, and it's an experimental viral vector for gene delivery protocols into host cells for therapeutic purposes. We talked about another helper virus earlier on in the course, and that was hepatitis delta virus that requires hepatitis B. So this is a second example of a virus that needs help to cause infection. Moving to slide number 23, parvovirus B19 serotype causes some infections. So virus needs those host cells that are dividing, continuously dividing host cells, including bone marrow, blood cells, and fetal liver. Virus replication causes the death of red blood cells. So even without telling you anything about the signs and symptoms or different types of the diseases that it causes, as long as you know it causes the death of the red blood cell, you can say, okay, red blood cell participates in oxygen delivery and basically providing nutrients to the tissues. So if you know that, and also know that if we have low red blood cells, we get anemia, different types of anemia. So if you know that, to some extent, you can perceive or you can postulate what kind of illnesses or diseases parvovirus B19 infections may cause. The good thing is that following infection, host immune response kicks in, so we develop immunity. Unfortunately, those patients who are immunodeficient, they have persistent infections, and persistent infections continuous death of the red blood cells results in anemia. Let's move to slide number 32. Human parvovirus B19, as I just mentioned in previous slide, infects mitotically active erythroid precursor cells in bone marrow. Okay, let's see what we're talking about. Erythroid precursor cells, those are precursors or the grandparents and parents of red blood cells or erythrocytes. These cells are highly active. They undergo mitosis a lot, and they can be found in the bone marrow in the red compartment or in the red marrow. Yellow marrow is fat. Red marrow is the site of blood cell formation or hematopoiesis. The diseases that are caused by human parvovirus B19 are erythema infectiosum, or fifth disease that we usually see it in children. Children will present mild flu-like symptoms. They have arthritis and they have rashes on their cheeks. So it's also called slapped cheek. It's like somebody has slapped these children in the face. And the second disease caused by parvovirus B19 is transient aplastic crisis, or TAC. It's a severe hemolytic anemia in patients who have other underlying anemias, such as sickle cell anemia, thalassemias, or acquired hemolytic anemias. So basically, these patients have an underlying genetic disorder such as sickle cell or thalassemia or acquired hemolytic anemias. And infection with parvovirus adds more onto the underlying cause. So basically, we end up with a much more severe hemolytic anemia, during which red blood cells are ruptured and patients suffer low numbers of red blood cells. The third disease caused by parvovirus B19 is called pure red cell aplasia, PRCA. It's a chronic anemia that is seen in immunodeficient patients. It's pure red blood cell. It means that the number of white blood cells and platelets are more or less within the normal range. Only red blood cells are affected. The fourth disease is called hydrops fetalis. That is a fatal anemia in fetus. However, there's a low risk of B19 infection during pregnancy, so we do not see a lot of cases of hydrops fetalis due to parvovirus B19. Moving to slide 33, summary. Adenovirus is an important pathogen that can cause a variety of diseases, including respiratory tract infection, conjunctivitis, gastroenteritis, and cystitis, or inflammation of urinary bladder. Adenovirus uses a viral DNA polymerase and a protein primer called terminal binding protein for continuous DNA replication of a parent strand. Remember, I mentioned that there is no discontinuous DNA synthesis during type 1 or type 2 DNA replication of adenovirus, but the hallmark is accumulation of large amounts of single-stranded DNA molecules. These are intermediates. So we see them for a short period of time because due to the presence of terminal inverted repeats, they will form circular single-stranded DNA structures. Messenger RNA is transcribed from both adenovirus DNA strands using cellular or host cell RNA polymerase II. mRNA splicing takes place to make numerous messenger RNA species, which will code for a myriad of proteins. Moving to slide number 34. Parvovirus B19 infects the undifferentiated erythroid precursor cells. These are the parents and grandparents of the red blood cells that are found in the red bone marrow. Diseases caused by parvovirus B19 infection are childhood rash or erythema infectiosum, and different types of anemias, transient aplastic crisis or TAC, pure red cell aplasia, PRCA, and hydrops fetalis. So this is the end of the audio lecture on lecture set number 11. Please go over the slides as you have your the audio recording on, so you can listen to it and go over the slides. And please make sure to send me an email if you have any questions or any areas are unclear to you. Thank you so much for your time, and I hope that you could learn something from this lecture. I will communicate with you via announcements part of the Canvas in case there's something that we need to discuss. Thank you so much. Have a good day.